Today, we're pleased to welcome Dr. Evelyne Maes, who is the senior scientist leading the proteomics platform at AgResearch in New Zealand. She obtained her Ph.D. in 2014 from the University of Leuven in Belgium. During her Ph.D., she worked on the development of both molecular and elemental mass spectrometry methods to detect low-abundant proteins in a wide variety of clinical samples, and studied the changes in protein profiles of blood-derived immune cells of colorectal cancer patients. With her curiosity to discover other research fields where proteins were central players, she found her way to AgResearch in New Zealand, a key research organization focused on serving one of the country's largest economic sectors, agriculture. In her current position, she applies her experience in mass spectrometry to lead several projects that study proteins and endogenous peptides in samples across the value chain, from soils and pastures to food, like milk and meat, to stimulated digestions and nutritional interventions in human cohorts. Additionally, she tries to drive further methodological innovation in the agricultural proteomics field. So thank you, Evelyne, for joining us today, and we're very excited for your talk. Let me just stop this screen share, and then you should be able to share your screen at any point.
Good morning, everyone, or good evening, and thank you for joining my webinar, and thanks, Catherine, for this very nice introduction. Before I start my talk on proteomics and really dive deep into the science, let's quickly introduce AgResearch, and we'll see if this works. All right, so AgResearch is New Zealand's ground research Institute for Agricultural Science, and what we try to do is drive prosperity by transforming agriculture. At AgResearch, we partner with the pastoral sector to identify and deliver innovation and create and increase value for New Zealand while enhancing environment and land use. Our proteomics team is based at Lincoln, but we also have colleagues at other hubs in Palmerston North, Hamilton, as well as Mosgiel. The science that we deliver at AgResearch, we try to deliver new knowledge and technologies that underpin New Zealand's pastoral agri-food and agri-technology value chain, which means that we really have an approach from farm to fork as we are trying to get the smartest, most sustainable farming systems completely through the most sought after food and bio-based products. So at AgResearch, we lead in 5 main areas. The first one is provenance, which is tracing the journey of a product from where it's grown to where it's sold or served. And this is really to ensure that the product is safe, that the product is of premium quality, and that it's attractive to the consumers. A second focus is environmental impact, where we really drive innovative research for pasture fertilization, as well as soil, water, and nutrition management. And this is really to improve practices on farm and reduce environmental impact. Third focus is land use, where we support transition from farming systems to multifactorial land use. The fourth one, which is climate change, where we are researching a lot of new technologies and farm practices, which really help agriculture transition to the future low carbon economies. And last but not least, we also do a lot of research on future foods, so foods that can have health benefits, but we also try to create new foods or desirable flavors and textures. And why is this food so important? Well, the consumer needs for foods are changing. So everyone wants the healthiest foods, nice fresh products, colorful platter. It needs to have a lot of nutritional value. It needs to be ethically sourced. If we can digest it very well, that would be very helpful. We want to gain a lot of strength. And if we can stay healthy and be healthy by eating food, that is really what the consumer likes as well. So what I thought I would do today is really give a snapshot of a few of our projects where proteomics is involved in. And this is really from the farm to fork approach. So the first study I will explain a little bit more in detail is really on dairy cows. So in New Zealand, dairy or milk industry is quite a big industry. And as we know, milk comes from the dairy cows. So, and these cows have a lactation period of around 10 months. And while in many countries, these cows stay inside during lactation and only go on the pasture in the 2 months of their so-called dry periods, in New Zealand, these cows stay outside on the pasture most of the time. So it has a little bit of different farm management system. Also, we really aim the calves to be born in early spring, because then there is a lot of grass and fresh grass for the calves to eat and to grow quickly. So there's a little bit more pressure on how successful in artificial insemination in these dairy cows is. And we will really feature how proteomics can contribute good insights in this field. A second topic I will briefly touch up in is how we are trying to meet consumer preferences. And I will feature one of the iconic export products of New Zealand, which is lamb meat. And we really want to know whether or not our proteomics and peptidomics can generate insights in how consumers prefer certain types of meat. And lastly, I will also briefly touch upon some functional foods. So foods that keep us healthy and make us healthy. And although we typically would think of these as very colorful fruit or veggies, in this project we really look into whether or not meat can also be one of these potential functional foods. So the first project is the work of one of our PhD students, Nicolas Aranciaga, together with his supervisors, Jess, Jim, and Debbie, and also with Lincoln University and Darien Z. And he's really using proteomics and metabolomics in the search for biomarkers of cow uterine suitability in early pregnancy. So as I already mentioned, dairy is really important industry in New Zealand. And for dairy cows to maintain their lactation, they must produce calf every year. And since we know that around 30% of the pregnancies are lost in the first week, this reflects to around 500 million loss a year in New Zealand dairy cows. Early embryo in dairy cows is completely dependent on uterine fluid for nourishment. And because this is so important, the main topic of Nicolas' PhD is to understand what the links are between embryo quality and the molecular composition of uterine fluid. So in the design, what he did was having saline infusions and flushings of the cow uterus. And this is really twofold. So first to get the embryo after 7 days after insemination. And this embryo will be graded. We check whether or not it's viable or inviable. And in this study, there were 55 viable embryos and 28 inviable embryos. Next, this uterine fluid will be separated by molecular weight, and both proteins and metabolites are both proteins and metabolites are separated. We perform proteomics on high resolution LC-Q of MSMS and metabolomics via GCMS. And we use P6 Plus for the identification as well as labor-free quantification of the data. And afterwards, we use these data to be integrated in a systems approach. So if we look at a proteome analysis, one of the key things or the key difficulties of uterine fluid is the protein identification, because there are a few proteins that are very high abundant. It's almost similar as plasma in humans. So there are a few really high abundant proteins, and then the real proteins of interest are just of medium abundance. And in this protein identification, P6 Plus really helped us quite a bit, because we used the in-course approach where both P6 as well as mascot data were combined. And in the end, we could identify 1,335 proteins. And of these, 144 were not previously reported in uterine fluid. Next, we also did a label-free quantification approach on all these samples, and this quantification is based on the MS1, where we used the top 3 unique peptide areas. For our quantification, we only use proteins that are detected in at least 50% of the samples, and we ended up with having 781 of these proteins. if we look at the partially squared discriminant analysis of these proteome profiles, what we could see is that, unfortunately, just based on these profiles, no clear distinction could be made between the viable embryos and the unviable embryos, or the uterine fluid where these viable or unviable embryos were living in. So because there is no full separation achieved, what we could see is probably there's a lot of other variation present in this proteome, because there are differences in age, in milk production, and so on. So these differences might not be really related to embryo viability. And although we had all these variables, there's always a risk of overfitting. So what we did was using a multi-omics approach, and we used our 781 proteins that were identified and complemented with our metabolomics data, where there were 122 metabolites data. And combining those two made a completely different story. If we look at the partially square discriminant analysis here at the bottom, what we can see is that based on the combination of proteins and metabolites, there is a distinction possible between viable and unviable embryos, and we found the 48 biomarker candidates for embryo development. If we look at the loadings, which are the molecules that are the most important for making this distinction between viable and unviable, then we can see that although the top one is a metabolite, there are quite some proteins there. So it was really good to see that our proteins are really driving this process as well, but it's only with complement of metabolites that really this strong association is popping up. We did some pathway analysis via an overrepresentation analysis, and what we could see is that one of the biological pathways that was really popping up in this difference is chaperone-mediated. And this is not really surprising, because we kind of would think that stress is very important in the distinction between a viable and an unviable embryo. But yet again, it's really good to get this confirmation that some biological processes are ongoing there, and we can further dig into what this means from a biologic and farm systems perspective. So in conclusion of this study, what we can say is that Peak6 really helped us optimizing or maximizing our number of protein IDs. We had very high sensitivity in our quantification, and it's only with a systems approach of combining proteins and metabolites that we really can push forward and now have the 48 biomarker candidates for embryo development. A second case study I want to feature today is linking flavor to consumers via omics, and this is the work of Enrique Panvan together with Yang Fang Ye, Carolina Riela, and Shantanu Debshudhuri. And what the main topic of this project is really trying to deliver customized meat products and trying to understand how both New Zealand and Chinese consumers like the commercial lamb. And in this study, we had lambs that are raised on grass, on chicory, or on mixed pasture, and this is grass and clover. And the main question here is, can the feed of the animal influence the taste of the meat? And if so, can we link this to consumer preference? So before going into detail, so flavor is a multifactorial experience involving olfactory, gustatory, texture, mouthfeel, and an integration of all these senses. So I think that everyone knows the feeling of if you smell some food and it doesn't taste or it doesn't smell nice, then you wouldn't be tempted to eat it. Same as if you eat something that's very sour or very bitter, then it's also something where it's like, I'm not going to eat that very soon, so it's not really attractive. And while for taste, we all know this very typical representation of the 5 tastes on a tongue, unfortunately, it's a little bit more complex But what we do know is that the taste is very much linked to the molecules which are in the food. And these can both be metabolites such as lipids, aromatic compounds, and volatiles, but it can also be peptides. And the taste properties of these peptides have been shown to be dependent on the length and the strength and the sequence of these amino acids in these peptides. So what we did in this study is we had these 3 groups. We had 8 replicates per group, grilled the meat, and then took the centerpiece of the meat, grinded it to powder, and then extracted peptides from the powder. We centrifugated, ultrafiltrated, and then did an LC-MS in high resolution, identified the peptides, and then analyzed these data a little bit further in our in-house flavor database. So looking at the peptide analysis, what we could see is that overall there were in these 24 samples, we could identify 8,378 peptides at an FDR level of 5%. Reason why this FDR percent is a little bit higher is because we are looking firstly at smaller peptides, so the minimum length is 4 amino acids. On the other hand, this is a very unspecific digest, so we want to open up this false discovery rate a little bit. Looking at the modifications found in Peaks PTM, what we could see is that grilling didn't give us many really funky modifications, which is good because that's what we are looking for in this study. And from this 8,378 peptides, we could find 8,035 sequences, which were relating to 276 protein groups. Doing the taste analysis, we used our flavor database, which is an in-house database that is based on both peptide sequences as well as scientific literature, and it works in a twofold way. We can estimate the bitterness as well as we can really check whether or not there are certain taste peptides present as an exact match in the database. So if we look at bitterness, bitterness is typically related to the hydrophobic amino acids, so leucine, isoleucine, proline, phenylalanine, tyrosine, and tryptophan. And if you look here at the top panel, what we can see is that for the 3 groups, so the lambs raised on chicory, the ones on mixed pasture, and the ones on grass, no real differences are seen in both the number of peptides that were identified and also the number of bitter peptides that were found. As well, we did a evaluation of the Q value. Q value is really also a value on the hydrophobicity of the sequence of the peptide. And yet again, same as just the number of bitter peptides, we do see that there is definitely presence of bitter peptides, but there is no major difference between the 3 groups. If we then look at the sequences and compare this with our database and look for exact sequence matches, what we could see, and that's quite exciting, is that in one group, only in the ones that were raised on mixed pasture, we have umami peptides. As we know that flavor and taste is related not only to peptides, but also to metabolites. Also some metabolomics was performed. And in this project, we did analysis of the polar compounds in positive and negative mode, as well as the lipids in positive and negative mode. And if we look at all these metabolites together, the profiles really clearly show that there is a distinction between the 3 groups. And there's a little bit of a mix between the grass and the mixed pasture, which kind of makes sense because the mixed pasture is grass as well as clover. But there's a quite good distinction with the chicory data. If we look at the loadings that influence this difference between the groups, what we can see is that there are a lot of amino acids, which is also not really a surprise because they are quite good in activating taste receptors. And there are lots of organic compounds such as creatine, taurine, and so on and so forth. So if we want to link this to the consumer, what we do know is there's no difference in bitterness detected between the different postures, but there's definitely bitter peptides present. What we also know is that in the mixed postures, there are some umami peptides, and the characteristics of these umami peptides is really that it's a long-lasting taste that has some mouthfulness, very complex taste as well. And this will be very interesting because these data will be confirmed by a sensory panel, and a sensory panel in are people that are just tasting all these different samples without knowing what the background is, and they really in detail describe the taste or describe how they experience and the food and what their sensation is. And it's really based on these data of the sensory panel that we get more insights in what the consumer preference is, and this way we can of course also combine these things and get more insights. So the conclusion of this work is that the use of omics technologies to understand how food compounds contribute to taste and sensation is critical to understanding how consumers experience flavor. The last piece of work is one where I'm involved in together with my colleagues Stephen, NC, and Stefan, and it was a fantastic collaboration with Brugger, especially Michael Krawitzki, as well as BSI, especially Jonathan Krieger. And this work is a little bit more on functional foods. The work is also presented at ASMS Reboot, so for people that want to see the poster and have linked up to ASMS, definitely have a look there. And our question was whether 4D peptidomics could enhance the identification of bioactive peptides in meat hydrolysates. If we look at meat in general, we wouldn't really immediately link it to a functional food, but meat hydrolysates, which are made by the degradation of proteins using commercial enzymes, is already used quite often as a sort of flavor enhancers, emulsifiers, and nutrients. And we typically can see it as a meat powder. So it's, if we would buy it, it would be more in these type of formats. So what our question is maybe these are a really good source of bioactive peptides. And what are bioactive peptides? Peptides that are typically short, up to around 20 amino acids. They are generated from proteins, either during gastrointestinal digestion or prior to consumption, and they must have health-promoting activities in vivo. So they can be anti-hypertensive, hypertensive, immunomodulatory, antioxidant, antimicrobial, and so on and so forth. But by eating it, we should get health benefits. That's the idea behind functional foods. So our peptidomics workflow was the following. So we had some beef meat, we grounded it into powder, and we produced a hydrolysate. From this hydrolysate, we made an ultrafiltrate where we had all these endogenous peptides and then had an aliquot of around 100 microgram of peptides. We performed a high pH reverse phase fractionation into 12 peptides, 12 fractions, and then had these 12 fractions measured in data-dependent acquisition on a timsTOF Pro. Next, we used Peak6 Plus to really build a library of these 4D peptidomics data. And then we also trialed and see whether or not data-independent acquisition would help us even further with getting more insights in a very consistent way. Yet again, we use Peak6 Plus to identify these data, and then we link this to our in-house bioactivity database. By building this library in X6 Plus, we had one point where we had to take extra attention, and this is because we know the hydrolysate is produced by the use of pepsin, so we are expecting very short peptides. This also means that singly charged peptides needed to be included. And as you can see here from our library, the singly charged peptides are the most abundant one in our sample as well. We built a library at an FDR of 5% at the peptide level. Yet again, this is quite broad because the digestion is quite unspecific, and we are looking at smaller peptides as well. So the peptides are of the smaller length of only 4 amino acids long. If we look at the amount of matched ions per peptide, then we also can see that our library is of very good quality. And in this library, we ended up with 8,378 peptides that were identified. And in comparisons to our previous 3D acquisition, where we analyzed the same samples, we ended up with 1,810 peptides. So what we can see just from building this library is that there is a huge potential in analyzing these type of samples with 4D peptidomics. Next, we investigated whether DA-PASEF could help us even further and see whether or not this complete data completeness would generate more insights in many of the samples. So what you can see here is quite a typical view when someone would perform a DIA-PASEF experiment. What is a little bit atypical is that because we are interested in the 1+, we are also selecting these ion cloud, and it's maybe better if you see it in one of the views of the peak software, where it's really clear that there's a lot of 1+ features that are of interest in our analysis as well. Looking at some of the preliminary data here is that we did 3 injections on short gradients, and after a library search, we could find 2,618 peptides at the 1% FDR level. And although this might sound not as much as what I initially would have thought, if we compare this to the previous data we had, where the LC gradient is 3 times longer, we still see that we gain a lot of peptides. However, the really important or the really interesting part here is that when we do a bioactivity search, that we use our in-house database, and we look for both exact matches, which means that the sample contains the whole bioactive peptide that are found in the database, or a partial match where the sample contains a portion of the bioactive peptide, and this portion or this portion is smaller than 5 amino acids difference. If we then do a bioactivity search, what we could see from this data is that we found around 75% more peptides with predicted bioactivity when we apply 4D peptidomics. So although we know we need some longer gradients for this DIA approach to deliver the really crucial insights, this preliminary study really demonstrates the value of the enhanced 4D data acquisition to characterize bioactive peptides present in potential functional foods in much more depth than previously possible. So we're very excited about these data. And with this, I think I very much would like to acknowledge everyone from the proteins and metabolite team at AgResearch, just for their support, but also for their enthusiasm in all the work that we do in our Proteomics platform, as well as all the collaborators on this side. And if you would be interested in learning more, feel free to visit our website, and I'm happy to take any questions. Thank you.